Morning, everyone, and thanks for dialing in this morning. It's a real pleasure to have Nicole Gruta give our seminar today. If you don't know Nicole, you're in for a great ride about everything about T cells, especially how they might dysfunction in the elderly. I think that's Nicole's latest view on things, and that's a really important topic considering what we'll seeing in the news on a daily basis. So just to give you a very brief introduction, Nicole completed her PhD at Monash University, being born and bred in Melbourne, as she just shared. And then she went for a brief postdoc to St. Jude's Children's Hospital and returned to Melbourne to join Peter Doherty at the University of Melbourne in 2002, where she spent a very long time with Peter and the very successful team there. And in 2016, she became a professor at Monash University and currently holds an ARC Future Fellowship. So she's achieved everything that I ever want to do. So Nicole, it's a real pleasure to have you here, and we're looking forward to your talk.
Thanks, Anja. It was a pleasure to be invited. So I hope to be able to come up and visit you all in person, but that was not to be because we're all an infected mob down here in Melbourne, although not anymore. So what I'm going to be talking to you today is sort of two stories, one of which is what Anja touched on, which is, I guess, how T cells change with age. But the main story here is unpublished work that's currently been submitted, and it's really looking at what we've done over many years, trying to understand how the nature of the T cell receptor recognition of peptide MHC really informs the signal that the T cell receives. So just to, I guess, introduce CD8 positive T cells, and I'm sure it doesn't need any introduction to this crowd, but what we know is that CD8 positive T cell immunity is critical for immunity to viruses and other intracellular pathogens, and in fact for a number of important human diseases. It's the T cell responses that are the best correlative protection from these diseases. We know also that CD8 T cells can actually mediate a number of autoimmune diseases such as type 1 diabetes and multiple sclerosis. And we know that CD8 T cells are the focus of current cancer, the primary focus of current cancer immunotherapies. In fact, CD8 T cells are responsible in a steady state for eliminating transformed cells as they arise. But of course, current cancer immunotherapies are really focused on trying to, by and large, boost the signal, the activating signal that the CD8 positive T cell receives. Okay, so in order for T cells to perform all of these functions, they have to first be activated. And this activation occurs, so here we've got a naive CD8 positive T cell. And this activation occurs through recognition by the cell surface expressed T cell receptor of a peptide MHC complex on the surface of a professional antigen presenting cell. So here we have the self MHC bound to, for example, a viral peptide. Once that recognition event occurs, the signal is transduced. So also augmented by the co-receptor shown here, CD8, which also binds to class 1 and brings in the Sark family tyrosine kinase to the CD3 complex. So it's important to note that the TCR alpha beta heterodimer has no intrinsic signaling capacity, but must associate with CD3, which is then phosphorylated and signal transduction can occur. So once this signaling event takes place, the CD8 positive T cell becomes activated, and there are two main consequences of that activation. The first is proliferation, so the T cell expands in number, and the second is the acquisition of effector function, namely the ability to produce cytokines and the ability to deliver a direct cytotoxic hit to cells that present this same peptide MHC complex. So for example, on the surface of a virally infected target cell, the CD8 positive T cell can lyse this cell and in doing so can eliminate virus factories. So just to give you sort of, I guess, an overarching schematic of what we work on in the lab, it's really basically lots of different characteristics that or parameters that control the robustness or the strength of the CD8 positive T cell response. We've done a lot of work looking at the abundance of epitope-specific naive T cells, so essentially the arsenal that's available to recognize pathogens when they're encountered. We've done a lot of work, or quite a bit of work, looking at antigen abundance, and most recently we've done that work looking directly at epitopes that are viral epitopes that are presented by MHC via mass spec analyses, and this was done in collaboration with Nathan Croft and Tony Purcell. What I'm going to be talking to you about today is our work looking at the nature of the T cell receptor recognition of peptide MHC and how that informs T cell activation, but also intrinsic molecular determinants and in particular metabolic determinants of CD8 T cell functionality that change with age. Okay, so why are we interested in understanding TCR recognition of MHC? We know T cell receptors have to recognize peptide MHC and then the T cell becomes activated, but it's not that straightforward. And what we also know is that many current immunotherapies that I alluded to earlier rely on the augmentation of the signal that's received by the T cell after antigen recognition. And optimized T cell receptors have shown some success in the treatment of advanced myeloma and sarcoma, and also in the control of chronic viral infections. And by optimized, at this point, what I mean is targeted mutations within the T cell receptor to increase the strength of binding between the T cell receptor and peptide MHC. And these potent T cell receptors are being actively developed by major immunotherapeutics companies. And so on the back of this, I would argue that a comprehensive understanding of what constitutes effective T cell receptor recognition and how that then translates to signaling to the T cell are essential, both for our fundamental understanding, but also for clinical applications downstream. So looking at the T cell receptor, I mean, I guess with this slide, what I wanted to point out is that there are really two, I think, really unique features about this receptor ligand interaction. Over here, which I'm just showing you how the T cell receptor is generated. So this is the alpha chain of the T cell receptor, and this is the beta chain. And so both of these chains are formed through genetic recombination of different gene segments. In the case of the alpha chain, it's VG, variable joining and constant regions, and the beta chain has an additional gene segment, this D gene segment. So the T cell receptor chains are characterized by three regions of hypervariability. These are the CDRs 1, 2, and 3. But while the CDRs 1 and 2 are encoded by the V gene element, the CDR3 region sort of encompasses this junctional region where these different gene segments come together. And so these three CDRs actually correspond to these three sort of protein loops that dangle down and are the main points of contact with the peptide MHC. So I guess the two really unique features of this interaction is firstly that the T cell receptor has to recognize simultaneously both self and non-self. So the self MHC class 1 molecule and the non-self peptide, which is typically a virally derived peptide or perhaps a neopeptide. The other thing that really sort of characterizes this interaction is the incredible diversity. And so I mentioned to you how diversity in the TCR is generated, and this results in a theoretical diversity of around 10 to the 15 to 10 to the 20 different possible T cell receptors. But of course, in actuality, it's more like around 2 million in mice and around 25 million different T cell receptors in humans. But apart from TCR diversity, the MHC is highly polygenic and highly polymorphic, so multiple different alleles for each MHC. And of course, the array of potential peptides bound by the MHC is virtually limitless. So there's this incredible diversity that this interaction needs to navigate. So with that in mind, if we now, what this is showing you is taken from a review written by Jamie Rossjohn, and also this figure was created by Stephanie Graham. What it shows you is the MHC class 1 restricted T cell receptors for which there was a ternary structure of the T cell receptor interacting with the peptide MHC. And what you can perhaps see from this is that there is substantial variation in the docking angle of the TCR. There's variation in the buried surface area, so the extent of contact between the T cell receptor and the peptide MHC. And there's variation in the relative contribution to binding of the various elements that are involved in the interaction. But what's perhaps more interesting and surprising considering this diversity is what's conserved in this interaction. So almost without exception, the T cell receptor binds over the top of the peptide. So now we're looking down from the T cell receptor onto the peptide MHC. And in the grey blob there is the peptide. We know that the peptide's always contacted by at least one of the CDR3 loops, and typically both. So either the CDR3 alpha or the CDR3 beta. And most pertinent to what I'm going to be talking about today is that the T cell receptor always adopts a consensus polarity over the peptide MHC. And you can see this here, that the T cell receptor beta always docks over the MHC alpha 1 helix, and the TCR alpha always docks over the MHC alpha 2 helix. So this really highly conserved docking polarity has sort of driven I guess the generation or the development of two models to essentially explain MHC restriction. So why do T cell receptors have to recognize MHC? Why can't they recognize anything? The first model is the germline model, and that suggests that there is over millions of years of coevolution of the T cell receptor with MHC, that there are now sort of complementary regions between the T cell receptor and the MHC that sort of mandate that the T cell receptor recognizes the MHC in this highly conserved orientation. And that if it doesn't adopt this polarity over the MHC, that this would prevent significant binding of the T cell receptor to the MHC molecule. The alternate model, the selection model, suggests that in fact there is no intrinsic capacity of the T cell receptor to recognize MHC, but in fact the MHC restriction is enforced by the role of the co-receptor. That is, CD8 needs to bind the alpha-3 domain of MHC, and in doing so brings LCK into proximity with the CD3 complex. So there's lots of evidence both for and against each of these models, but I guess one thing that we can say is that while the germline model clearly sort of stipulates the polarity with which the TCR needs to dock over the peptide MHC, the selection model at this point doesn't. That was until the last few years. So this was a while ago. In 2012, Roy Mariuzza published this ternary structure of a T cell receptor in complex with MHC class 2 and the CD4 co-receptor, which essentially has the same role as the CD8 co-receptor. And he found that it formed an arch-like structure, and he suggested that this arch-like structure facilitated the interaction of LCK with CD3, although where these molecules sat was not clear at this point. And then last year, in a really seminal paper, the structure of the T cell receptor in complex with CD3 was resolved. And what you can see here is that the CD3 complex actually sits to one side of the T cell receptor. And they've used Mariuzza's model here in the paper to show where the CD3 might sit in relation to the CD4 co-receptor, showing that they are proximal to one another. And so now if we revisit the selection model, with a sort of axially asymmetric arrangement of CD3 around the T cell receptor, what you can see is that a canonical or conventional docking polarity of the T cell receptor might result in co-localization of LCK and CD3, where a reverse orientation of the T cell receptor might place CD3 outside of the arch and therefore too distal to LCK to facilitate effective signaling. Now, we really haven't been able to address this because, as I said, this docking polarity that we observe is highly reproducible. And until recently, we've not seen anything that sits outside of that arrangement. And that's sort of where our work comes into the story. We use a Black 6 mouse model of influenza A virus infection, and we're interested in looking at the CD8 T cell responses to the various influenza epitopes. Of note is this CD8 T cell response, which is immunodominant, which means it makes up a large part of the CD8 T cell response to flu, and it's targeting this NP366 to 374 epitope within the nuclear protein of flu. And it's this NP epitope bound to the H2DB MHC class 1 molecule. If we look at the T cell receptors that recognize this NP-DB complex, what we know is in the immune repertoire, it's characterized by this TRBV 13.1 prominence. So that just means that a majority of the T cell receptors that recognize this epitope use the TRBV 13.1 V gene segment in their beta chain. If we look at the corresponding naive repertoire, so these are naive T cells that are specific for the DB-NP complex, you can see that this 13.1 bias is still present, although at a reduced prevalence relative to the immune repertoire, suggesting these cells are preferentially expanded into the immune response. But what was of particular interest was this TRBV17 positive subset. So this is multiple different T cell receptors, but they're all using this TRBV17 beta chain. And what we found was that they were almost never seen in the immune response. So this got us interested in, well, why would you have a quarter of your available repertoire that's simply not being recruited into the immune response? So at this point, we collaborated with Jamie Rossjohn at Monash University and got a crystal structure of 2 of these TRBV17 positive T cell receptors in complex with the DBNP peptide MHC class 1 molecule. And what you can see here, this is just a canonical, it's not really very informative, it's just a canonical T cell receptor recognizing an peptide MHC class 1 complex. And this is just to show you that it binds in a canonical orientation. But what we found was that these 2 TRBV17 TCRs that we identified from the NP repertoire bound in a completely reversed orientation. So they were 180 degrees flipped around. So now you can see that the beta chain docks over the alpha 2 helix and the alpha chain docks over the alpha 1 helix. This was sort of note, I suppose, because it was the first time TCRs had, the structure of TCRs in complex with peptide MHC had been solved for TCRs from the naive repertoire, so the unexpanded repertoire. And it suggested that there may be a breadth of recognition modalities in the naive repertoire that aren't utilized in the immune repertoire. So now we were in a position to really try to understand perhaps almost even a bigger question. So is this canonical TCR recognition of MHC essential for T cell activation? Because we didn't even really know that. And if it is, then why does the T cell receptor have to recognize peptide MHC in this orientation? So what I'm showing you here was published in that first paper. It's the sort of the docking footprint of this this reverse T cell receptor over the top of the peptide MHC. We've made individual point mutations here and then mapped which of these mutations actually reduced the binding affinity of the interaction. What I'm showing you in black means that when these residues are mutated, that had no impact on the binding affinity. In orange, these residues had a moderate 3 to 5-fold decrease in affinity, and in red, these residues were critical for the binding. So when we knocked them out, there was the binding was essentially gone. So what was interesting, firstly, is that this interaction didn't use the typical interaction. So position 65, 69, and 155 are 3 residues that are almost always contacted by MHC class 1 restricted T cell receptors, and they were dispensable for this interaction. The TCR bound right at this carboxy terminal end. But what was most interesting was these 2 residues, this E18 and A89, because they had never previously been found to be contacted by a class 1 restricted T cell receptor. When we investigated these residues further, so we made a range of different mutations at these positions, you can see that mutating these positions had no impact on canonical docking T cell receptor. I should say this is a canonical T cell receptor expressed in vitro, and then we're looking at tetramer binding here. So we can mutate these residues. It doesn't have any impact on the ability of these T cell receptors to bind. But if we mutate those residues for the reverse docker, you can see it does reduce the binding capacity. And in the case of the A89E substitution, it completely ablates the ability of the reverse docking T cell receptor to bind, but doesn't impact the canonical docker. So now we had the ability to create a tetrameric staining reagent that could differentiate between canonical docking T cell receptors and reverse docking T cell receptors. So we use that to look at these NP-specific T cells in the naive repertoire firstly. And all we've done here is pulled the cells from 10 mice, and we've looked at the TRBV13 positive T cells and their ability to bind tetramers. And I told you these were the T cells that were really well recruited and expanded and dock in a conventional orientation. You can see relative to wild type tetramer, the A89E tetramer is detecting the same proportion of these TRBV13 positive T cells, suggesting that all of them dock in a canonical orientation, or at least don't require this A89 residue. Conversely, when we look at the TRBV17 positive cells, you can see that we're losing a significant proportion of these cells when we stain with the A89E mutant tetramer, suggesting that it's not just these two T cell receptors that are docking in a reverse orientation. We think it's at least 50%. We now know that this is an underestimation, because we know that if reverse dockers bind with a very high affinity, they can overcome this requirement for this A89 residue. Interestingly, if we look in the immune repertoire, so after we've infected these mice with flu and the T cells have expanded, we can now see that none of these reverse dockers can get into the immune repertoire. Okay, so more evidence that the reverse docking prevents these cells from getting into the immune repertoire. And initially, and this was published in the first paper, we thought that this might have had something to do with affinity. If we look here at the level of T cell receptor, and we gate for high, intermediate, or low T cell receptor expression, and then we stain with tetramer, you can see that the canonical dockers here bind more tetramer for a given level of T cell receptor than the reverse dockers. And this is shown just graphically here, where we expressed a number of different canonical TCRs and the two reverse TCRs. And you can see they don't bind tetramer as well, suggesting they have a lower avidity for tetramer. And so we put it down, the poor recruitment, down to the fact that, well, they're probably just not getting a strong enough signal because they don't bind as well. But we know that the normal range of affinities is 1 to 100 micromolar. And these T cell receptors bind with around 40 micromolar. So, you know, yes, it's a moderate affinity for peptide MHC, but it's not outside of the realms of what we know can support T cell activation and proliferation. So we decided to investigate this further. So this is just to show you what we've done here is look at NP specific cells, either TRBV, this is TRBV13, so you can see this is expanded, or TRBV17. If we gate on the TRBV17 cells and we look in the naive repertoire and the immune repertoire, really all I want, and what we've done here is sequence the T cell receptors, but really all I want you to get from this is that you can see that there's a lot of different naive T cell receptors that are available, but typically only one of them is, you know, robustly recruited into the immune response, which is really just reinforcing what I told you earlier. But you can see this T cell receptor is derived from a single T cell and it's expanded. So if I show you that graphically, here again, we've got a range of different TRBV17 positive T cell receptors that are available in the naive repertoire. When we infect, only one of them can really be recruited. So we took this blue T cell receptor and we expressed it in vitro. And what you can see is relative to a canonical T cell receptor, the TRBV13, this binds tetramer really poorly. So for a given level of T cell receptor, you've got much less tetramer binding than the canonical docking T cell receptor. Conversely, this T cell receptor that was very poorly recruited into the immune response binds tetramer just fine. So this was interesting. So then we got the crystal structures on these T cell receptors and also the SPR, so a measure of affinity. And what we found was that this reasonably well expanded T cell, the T cell receptors here docked in a conventional orientation with an exceptionally low affinity. Whereas this T cell that was poorly recruited docked in a reversed orientation, but had a very high affinity. And so this was really the first evidence that the reverse docking of the T cell receptor was preventing T cell activation in a manner that was independent of the strength of the T cell receptor peptide MHC interaction. So we then tried to look at this in vivo by making retrogenic mice. So these are retrogenic mice expressing, the T cell receptors of interest. Here we've got canonical docking T cell receptors of very high affinity or low affinity, and the reverse docking T cell receptors, the moderate affinity and the very high affinity. And really, so what we've done is we transferred these either in combination with one another or individually. And what we found was reproducibly that only the canonical docking T cell receptors could be recruited into the immune response after flu infection. That's shown in blue and pink here. But the reverse docking T cell receptors could not support efficient recruitment and expansion, irrespective of whether they were high affinity, which is shown in the light green here. So again, irrespective of affinity, reverse docking T cell receptors can't support recruitment and activation. So that brings us back to our models here. So, okay, so we've definitively shown, I think, that the reverse docking can't support recruitment and activation. So why can't it? And so here we are back at our model where I guess the hypothesis is that when the T cell receptor docks in a reverse orientation, CD3 is now too far away from LCK to be efficiently phosphorylated. So to test this directly, I'm going to skip. This is just modeling that we did, looking at where the reverse docking polarity might position LCK. And so we use that, the CD4 class 2 model of Mariutz's to try to model this. And you can see it really poorly positions the co-receptor relative to CD3. But here what we did is look at this directly by using a system that had been established by Mick Gascoigne at NUS in Singapore, where we transduce cells with a T cell receptor of interest. So this is the canonical high affinity T cell receptor. And we also transduce them with a CD3 zeta GFP fusion protein and a CD8 beta M cherry fusion protein as a proxy for LCK. And what you can see, and then we stimulated these cells with dendritic cells that were either pulsed or not with peptide and looked at flim fret. So basically the co-localization of GFP and M cherry is indicated by a loss of fluorescence of the donor fluorophore. And here what you can see, this is graphed over here, is that at the regions where it's contacting the dendritic cell, you're seeing significant flim. And that's when we don't see flim fret when we don't add the peptide, but when we do add the peptide, we see a significant loss of this fluorescence. When we look at the reverse docking T cell receptors, irrespective of their affinity, we don't see any flim fret, and that's shown here when we add the peptide. And then when we look at these other low affinity canonical docking T cell receptors, you can see again, we're seeing substantial flim fret here, suggesting that it's only when the TCR adopts a canonical orientation that CD8 and CD3 are co-localized. So based on this model, this would suggest, if we just skip back quickly, to here, to here, this would suggest that it's the sequestration of LCK by the CD8 co-receptor that's preventing the signaling. So if we free LCK from the CD8 co-receptor, can we now see signaling in the reverse docking T cell receptors? And so that's what we've done here. Here we've got TCR transductants expressing wild type CD8 with a canonical T cell receptor or a reverse T cell receptor. And what you can see, as expected, is that the canonical T cell receptor can support really robust signal transduction, as evidenced by phosphorylated ERK and IL-2 production, while the reverse docking T cell receptor really can't signal detectably by phospho-ERK or by IL-2 production. If we take CD8 out of these transductants, you can see now we've really hurt the signaling by the canonical T cell receptor, but we've increased slightly the ability of the reverse docking T cell receptor to signal, and that's evidenced both by phospho-ERK and IL-2 production. It might be argued that the CD8 co-receptor might be sort of acting to sterically hinder the signaling by the reverse docking T cell receptor. So what we did instead was we kept the CD8 co-receptor in there, but we mutated it so that it could no longer bind the LCK tyrosine kinase. And so what you can see here is that we now get improved signaling relative to the CD8 null, because CD8 is binding to MHC and stabilizing that interaction. And still, both the canonical and the reverse docking T cell receptors can signal, as evidenced by phospho-ERK and IL-2. So, and this is just showing you that with the mutated CD8, you can see that CD8's in there, but it's not binding LCK anymore. And so really that's sort of the end of this story. I'm not going to go through, this is the data essentially that I just showed you, but we think this is really exciting because we think that, despite the incredible diversity, that T cell receptor recognition of peptide MHC is constrained to facilitate the co-localization of these key signaling molecules. And why would the T cell receptor want to do this? We think that excessive T cell receptor peptide MHC cross-reactivity, and we know that T cell receptors are highly cross-reactive, is constrained by the number of signaling competent binding modalities. And so this then in turn enhances the exquisite specificity of the TCR peptide MHC interaction. So you've got this inordinate diversity, which I think can allow for lots of different binding events. And that's shown here, where you've got the TCR binding in a reversed orientation or binding on the side of the peptide MHC. But it's only when the TCR binds in this canonical docking topology with the peptide MHC that you actually get signaling and a functional outcome of that interaction. So that's sort of the end of that first part of the talk. And so this work has been submitted and it's under review at the moment. And now I'm just going to change tack a bit and talk about our recent work looking at the impact sort of metabolic characteristics on CD8 T cells and survival, CD8 T cell survival and function with age. And this is going to be brief, and I hope not to bore you with this, but I'll just go through the key points. So essentially what we know occurs with aging is that you get a change in function and phenotype of CD8 positive T cells. So in young mice, you can see that what we have is a majority of the cells are actually naive T cells. And so in steady state mice and humans, I should point out that typically we understand CD8 T cells as existing as either naive or memory cells. So they either haven't seen antigen or they have seen antigen. But what's been recently discovered is that there's a population of cells that are antigenically naive, so they haven't seen antigen, but they exist in a semi-differentiated state and express markers of memory T cells. These cells are called virtual memory T cells, and they can be differentiated, so they sit here. They can be differentiated from conventional memory cells by the expression of CD49D. So memory cells are CD44 high, CD49D high, shown here, and virtual memory cells are CD44 high, CD49D low. And what's interesting is with age, you can see that the virtual memory population really increases dramatically, such that if we look in aged individuals, you can see that around 50% of their antigenically naive T cells are virtual memory cells, whereas in young mice, it's by far and away mostly true naive cells. This is also true in humans for the putative virtual memory population observed in humans. You can see, again, this population increases with age such that it makes up around 50% of antigenically naive T cells in elderly humans. So a few years ago, we did an analysis of, we know that there's a sort of primary T cell dysfunction, and we were interested then with this discovery of virtual memory cells, did the dysfunction lie largely with virtual memory cells or lie with true naive cells? And essentially what we found is that if we look at true naive cells and virtual memory cells, we did a single cell proliferation assay, and this is showing you the proportion of those single cells that are able to proliferate in response to anti-CD3 stimulation. And down here is the clonal burst size of each of those cells that could proliferate. So we can see at 3 months in a young adult mouse, both true naive and virtual memory cells are pretty well capable of responding to polyclonal stimulation. You should note that virtual memory cells, by virtue of this semi-differentiated state, are known to actually proliferate better than true naive cells. And this is possibly the purpose of this sort of semi-differentiated state. But what we know is that with advanced age, in fact, the true naive cells retain their function remarkably well, while the virtual memory cells became almost completely dysfunctional with age. And this is reflected both in the proportion of cells that are able to proliferate, but also in that clonal burst size. So of those cells that could proliferate, they proliferated very poorly. So, so if we go back to here, I guess with this, sort of relatively novel population of virtual memory cells, we were interested in trying to look at sort of, I guess, accepted paradigms of naive cells and memory cells that had been published in the literature. And one of these was sort of how the steady state metabolic, I guess, state of T cells really characterized their differentiation state. So for example, true naive cells, we know, are metabolically quiescent. They have a basal nutrient uptake, a basal glycolytic rate, and they principally use oxidative phosphorylation for their energy. When a cell becomes activated and differentiates into an effector cell, obviously the cells become much more metabolically active. There's an increased energy requirement for protein, lipid, nucleic acid synthesis. And so the spare respiratory capacity, or the oxidative phosphorylation rate, goes down, a massively increased glycolytic rate for very rapid production of energy to fuel cell proliferation and effector function. Of particular interest to us was the memory cell population. It had been suggested that these cells existed in a metabolically primed state. They had very high spare respiratory capacity, an increase in the mitochondrial mass, an increase in the mitochondrial fusion, so an actually altered mitochondrial morphology, and an increased reliance on fatty acid oxidation. And it was thought that this promoted both the cellular longevity, we know memory cells are very long-lived, and also the rapid functionality of memory cells after they're re-exposed to their antigen. Now, it wasn't just that these characteristics were associated with memory cells. I mean, there were various experiments that had been performed to show that it was these metabolic characteristics that imparted these sort of functional attributes on memory cells. And so we wanted to look at this now, taking virtual memory cells into account. So here, what I'm showing you is a seahorse assay where we can look at the basal oxygen consumption rate, we can look at maximal oxygen consumption rate, and the difference between these two parameters is the spare respiratory capacity, which is thought to be a key marker of memory cells. And the first thing we really noticed, if you see the filled circles here are young cells from young mice, is that memory cells in fact do not have a high spare respiratory capacity relative to naive cells. In fact, it's the virtual memory cells that have an elevated spare respiratory capacity. For both memory phenotype cells, that is virtual memory cells and memory cells, this spare respiratory capacity goes up with age. So the spare respiratory capacity is increased. It doesn't change with naive cells. So right off the bat, this sort of showed us that there was no association between spare respiratory capacity and functionality in these T cells, because the most dysfunctional T cells basically are the aged virtual memory cells and the aged memory cells, both of which had very high spare respiratory capacity. And even in young mice, the spare respiratory capacity was very low in the memory cell population. Then, sorry, this is just quantitated here. So we know that changes in spare respiratory capacity often reflect quantitative or qualitative changes in the mitochondria themselves. So change in crystalline morphology, change in sort of overall mitochondrial morphology and mitochondrial footprint. So we tried to look at mitochondrial footprint just using confocal imaging, which was sort of satisfactory. We did see an increase in mitochondrial footprint in virtual memory and memory cells, and this tended to increase with age. Perhaps a better measure of mitochondrial load was looking directly at the amount of complex 4 in the mitochondria, which is part of the electron transport chain. And this was done by Luke Formosa in a collaboration with Mike Ryan's lab. And you can see that this is way up in virtual memory cells, and it's increased even further with age. So this elevated SRC did look like it was associated with an increase in mitochondrial load. Then we looked at the mitochondrial morphology. So it had been suggested that in memory cells, they exhibited this fused mitochondrial morphology, which essentially gave them these sort of superpowers to respond really rapidly after re-exposure to antigen. In fact, when we looked at true naive virtual memory and memory cells from young animals, and we quantitated sort of fused versus fragmented mitochondria, the majority of the mitochondria in all of these cell subsets was fragmented. Relatively little of it showed this fused morphology. And when we looked in aged cells at the mitochondria, certainly there was a significant increase in this fused mitochondrial morphology, but this really occurred across the board. And you can see that it's particularly prominent in both virtual memory and memory cells. And so, we deduced from this that this fused mitochondrial morphology was not intrinsically linked to this superior T cell function, and in fact is more likely to be reflective of a stress response that results in fusion of mitochondria sort of to enable sort of complementarity to compensate for dysfunctional mitochondria, which has been described outside of the T cell literature. So we just looked at, we'd done RNA-seq previously, and what we'd seen was, when we looked at these transcripts, is that transcripts that were associated with mitophagy, that is ATG101 and ULK1, were down with age across the board, suggesting that, with age, I should point out, suggesting that mitophagy may be inhibited with age. And also PGC1 alpha, which is a sort of master regulator of mitochondrial biogenesis, was significantly up in virtual memory and memory cells with age, suggesting that there might be a combination of effects, which is a reduction in mitophagy and an increase in mitochondrial biogenesis, maybe resulting in this increased mitochondrial load. So this figure is just to show you that we looked at a range of different T cell functions as opposed to just looking at proliferation, which I showed you earlier. And none of these functions, proliferation, cytotoxicity, or cytokine production, correlated with this spare respiratory capacity. The only thing that did correlate quite well was survival, so as evidenced by BCL2 expression. So it is possible that these mitochondrial changes are occurring to promote the survival of these, or the longevity of these cells. So I think, I think one of the major points of this recent paper is that here we have conventionally defined central memory T cells. They're CD44 high and they're CD62L high. And if we look within this population and look at how many of these cells are actually virtual memory cells in a normal mouse, so uninfected mouse, what we can see is that the vast majority of them, so over 80% of these mice in both young and aged mice, are virtual memory cells and not central memory cells. And even if we infect these mice with LCMV, which induces a really massive antigen-specific CD8 positive T cell response, still over 60% of, and this is 40 days after that LCMV infection, still over 60% of what we would have called central memory cells are actually virtual memory cells. So, I think there's a lot of scope here for sort of re-examination of characteristics that we have formerly attributed to memory cells and antigen experience cells, when in fact they may be a characteristic of virtual memory cells. So why this elevated SRC? So we know that virtual memory cells are highly sensitive to cytokines, and in particular IL-15. So what we did here was we did the seahorse assay on true naive conventional memory and virtual memory cells straight out of a naive mouse, but then we also infected mice and looked at memory and virtual memory cells at 2 time points after flu infection. So again, what you can see here is if we just take memory cells out of a normal mouse, this is what I showed you earlier, there's a lower SRC than true naive cells. Interestingly, even in the face of this response to flu infection, there's no change in the SRC of these memory cells. So many of these will be flu-specific memory, of course. By contrast, if we look at virtual memory cells straight out of a mouse, which again is what I showed you before, you can see there's an increase in SRC in the virtual memory population. And this has increased even further following flu infection. And this is interesting because by definition, these cells are not responding in an antigen-specific way to the flu infection. So they really almost have to be being affected by the inflammatory environment. So we looked then, because we know that they're particularly sensitive to IL-15, we did this whole experiment again, but introduced an anti-IL-15 neutralizing monoclonal antibody. And so what you can see here is this increase in SRC after flu infection in the virtual memory population, which is brought right down here when we introduce this neutralizing anti-IL-15 monoclonal antibody. So this indicates that it's sort of physiological levels of IL-15 that are driving up this spare respiratory capacity in the virtual memory T cells. We also showed, the dogma is that memory cell high SRC is being driven by fatty acid oxidation. This was actually shown not to be the case. 3 back-to-back publications showed that those studies had used levels of etamoxia, which inhibits CBT1, a rate-limiting enzyme for FAO, that the levels that etamoxia was being used at were far too high, and that in fact was shutting down the whole of the electron transport chain. And so what we found was that if we use these high levels of etamoxia, yes, we could really shut down this spare respiratory capacity. But if we used the lower levels of etamoxia, which have been shown to be specific for this fatty acid oxidation pathway, you can see that fatty acid oxidation really is contributing very little to the spare respiratory capacity of these virtual memory cells. We don't know what is driving this SRC in these cells, but it doesn't appear to be fatty acid oxidation. And so the reviewers asked us to look in humans. So here, what we've done here is look at, this is the IL-15 receptor beta chain expression in humans. The antibody is not very good, but you can see that we are seeing an increase across different T cell subsets with age in this IL-15 receptor beta expression. So their sensitivity to IL-15 is increasing with age. We couldn't do the seahorse analysis on the subsets because the seahorse analysis requires a very large number of cells, but we did it on total young CD8 T cells and total age CD8 T cells. And what you can see, it's not significant, but there is a trend toward an increased spare respiratory capacity in age CD8 T cells from humans relative to the young CD8 T cells. So as far as we can tell, these observations are holding up in humans as well. And so I'll finish there just to summarize that virtual memory cells preferentially accumulate in aged mice and humans, and they are the ones that exhibit the majority of this intrinsic proliferative dysfunction in the antigenically naive T cell population. So yes, spare respiratory capacity increases with age in memory phenotype cells, but it is in no way associated with T cell functionality. And in advanced age, this fused mitochondrial morphology occurs across all of the CD8 T cell subsets, and it could be associated with CD8 T cell survival, but again, not function. And it's the enhanced sensitivity to IL-15 that's driving this high SRC in virtual memory T cells. And so I'll finish there and thank the people who did this work in my lab. In particular, Peruse Ray is really driving the reverse stocking TCR story, and Kylie Quinn drove the aging T cell story. This is a collaboration with Mike Ryan's lab and all of the labs listed here, but in particular, I'd like to highlight Jamie Ross-John's lab. So the reverse stocking TCR story has really been a partnership with Jamie and Stephanie. And all of the platform facilities at Monash, I want to give a shout out to that have been invaluable. So thanks very much for your attention, and I'm happy to take questions.